Yeah, well, we'll have Trevor's presentation, and then we'll have a question and answer period, a general one for. Hold on. Are the observers going to be part of this?
Yes, so everyone was.
Okay, so we need to.
Online. This is open.
Okay, I don't.
So Trevor,
so
you're
not.
now presenter, so you can go ahead and present from your screen.
Okay, I don't have a PowerPoint presentation or anything, so I'm just going to speak, if that's okay. I do have notes, but I'm just going to speak from my notes rather than show them as a PowerPoint or something like that.
Right, that's fine. Thanks, Trevor.
So, hi everybody. This is a bit weird presenting to a screen where I can't see everybody's faces, but I'm an associate professor at the School of Aquatic and Fishery Sciences at the University of Washington, and I've been asked to come in and do an independent peer review of the management strategy evaluation process at IPHC. I did this by reading through all the documentation in the past and by attending one of the informal MSAB meetings, and by chatting to not an extensive group, but a selection of people that are from industry, government, and of course the MSE team themselves and people from IPHC. So really overall what I found was that the MSE is technically sound, follows international standards, it's coded for rapid evaluation using pretty advanced computer science techniques. It works well. The only minor thing I would point out there is that better documentation of the equations is needed, that the technical document for the MSE was incomplete. The performance metrics that were agreed amongst the MSAB cover all the standard evaluations that you'd like to see of an MSE, of harvest control rules and so on and so forth. Although I did feel there was a need for a dashboard-like interface where you could look at all the metrics in one place rather than paging to and fro to see the different impacts of different rules, harvest control rules, etc. The shiny website, interactive website, was a good idea. I felt like it needed a bit more documentation to get broader uptake from everybody involved. Overall, one of the uniform things that I found chatting to people was that everybody had high praise for the MSE team itself, and especially for Alan Hicks. They seem to be trusted by IPHC, government, industry, and all of the other stakeholders represented that I chatted to. That might not be true in general, but of the ones I chatted to. And a lot of people gave unsolicited praise for the process and the team involved. The MSAB board itself, I think, is a particularly strong feature of the MSE process. Usually the hardest part about MSE is gathering widespread acceptance of the process involved in coming to results. And I felt that the membership was well represented, different groups well, that people were committed to the process, that the various representatives of MSAB were well informed and committed to the process. And this is one of the best cases I've seen of MSE working in practice as a collaborative mechanism between industry, government, science to achieve results. The three things that I really felt needed a bit of work were, first of all, to resolve the uncertainty about where the MSE is going in the future. And especially this applies to the current MSE team, which really comprises three core members, Alan Hicks, Karpy, and Berikoff. And both Karpy and Berikoff are on contracts that are due to expire soon. And I really feel that the IPHC needs to decide if this is an ongoing process where they're going to be retained, and if so, plan and budget accordingly. The second thing was, instead of seeing the MSC as a process that's finished in January 2021 with a presentation for catch allocations, to see this as an ongoing process where every year you run the MSC on a different set of questions to ask whether the current stock assessment process is robust, to ask whether the current data gathering process is robust. So typical things that people have used MSEs for in other contexts are, for example, testing whether a different survey frequency would be okay, if the survey effort could be changed, testing whether the 4 assessment models are all needed or whether they should be weighted differently, testing whether the harvest control rule is robust to various important stock assessment parameters that are assumed, such as values for steepness or natural mortality or weighted age. So to see the MSC as a process where every year you will be answering a different set of questions, presenting those alongside with the stock assessment to ensure that everything is robust. The third thing, which certainly gets into the politics of how IPHC operates, is to codify how the Commission actually adjusts catches after the science is applied. So there's this one final step where the science comes out. It says this is recommended catch limits, mortality limits for each area, and then the Commission adjusts those to meet various needs of the participants in every area. And this final step is a step that's not included in the MSC process, but should be. And in other places where you have this kind of last minute changes to catches, things can go awry. So in the European Union, this last final step is where all the politicians sit and haggle over whether they should trade catches for increased agricultural products in Spain or whatever, and inevitably they catch too much. I don't think the same thing is going on in IPHC, but I do think it's important for the Commission to codify these last minute changes so that they can be represented accurately in the management strategy evaluations. So those are my 3 major things that I would like to highlight. There were some other minor points to think about. One of these is that if you're going to go into a situation where you use the MSC process to set harvest control rules, to decide on rules that could be used to set catches or mortality limits, you need to also specify under what conditions you're not going to do that. In other words, what are the exceptional circumstances under which you would stop doing the harvest control rule and restart the MSC process? And this could be things like, what if COVID stops the survey from running and you have no survey for 2 years? Then obviously, if your rule assumes you're going to have a survey every year, you suddenly don't, then you need to restart the MSC process. So deciding what exceptional circumstances might be and pre-specifying those before implementing anything. The second thing is that the MSAB membership could be expanded a bit to include other groups not currently included, such as crew members, communities, environmental organizations. Again, this is a minor point. And the third one I touched on already, which is that the technical documentation of the MSC needs to be updated and completed together with the results in January. So those are the highlights of the report. I do include a bit more in the formal report. I'm happy to answer any questions and receive feedback, and then I'll finalize the report for broader dissemination. Over and out.
Thank you, Trevor. So we can open it up to questions from participants, maybe starting with the SRB.
Yeah, I'll jump in here, Sean, if nobody else is.
Sure.
Thank you for the presentation, Trevor. This is Olaf Jensen. I wanted to follow up on something that you mentioned briefly in the written report, which was that the MSC could also be used to test the impact of different structural assumptions in the operating model. Could you elaborate a bit more on that and also give us some insight into whether structural uncertainties are already well represented in the operating model?
Yes, it is not to criticize the current assessment process, which already has 4 separate assessment models that does look at a variety of different assumptions to look at the impact. But what the MSE uniquely does is it asks, what is the long-term impact of getting something wrong? So for instance, if you have 3 or 4 harvest control rules that you can test that would set catches into the future, what would happen if you applied those control rules and the truth about, say, natural mortality was different to what you assume in your assessment model? So what if natural mortality was half what you think it is? How bad would your entire process be in the future? What is the long-term impact of getting something structurally wrong in the process? And the good thing about this is that, say you had natural mortality wrong, the assessment model would be a bit wrong. It would give a catch that would be slightly wrong, and then it would feed back. The stock might go down, which would feed back into the rule, which would say, oh, wait, we need to do something differently, and it usually would be robust to any mistakes like that. So what you want to test is that the whole process, the combination of the assessment, the data gathering, and the rule for setting harvest are all robust to getting something wrong at any point in the process.
Great, thank you. And one of the structural uncertainties that we've talked about in particular is the potential for density-dependent habitat selection. Do you think the MSE is set up in a way that could evaluate this?
Yes, I mean, I think almost anything you can think of could be modeled. It might be hard to model and get it right. But again, you could do something like where the assessment process, the 4 current stock assessment models, do not assume that, but the operating model in the MSE process does assume that, and you ask how wrong would the entire process be over several years under that scenario. So this is where I say that the MSE should be an ongoing process where every year, if that's the question that's on the forefront of the SRB this year, then next year the MSE process could run that scenario and ask whether the stock assessment process is robust to that scenario.
Great, thank you.
Sorry, just so I understand that, I think I have a very similar question. This is Sven Kupschus from the Bad Europeans. I just wanted to understand, it's always going to be conditional on the MSE or the operational model underlying this. So I find it difficult to see that you, under all circumstances, you would have to have a whole plethora of, operating models or potential operating models, versions of operating models, which I thought, or at least I got the impression, was quite difficult to do in this setup because it was so optimized for being quick and timely that in order to do MSE evaluations, they are steering the whole operating model through I think a text file or some sort of simplified input file. They both have benefits, and I just wanted to see where you think the risks or which side of the coin you come out at this.
Well, given that I have a computer science background far in the distant past, I'm of the opinion that if you can think up a scenario, then somebody can program it up into an operating model. It might be hard to program up. It might take time, but everything is, in my mind, everything is possible.
Thank you.
I guess we're having similar veins of ideas here, but I had a couple of questions that maybe can integrate into one, but I was going to ask whether you had some suggestions or if you had noticed any potential for prioritizing potential operating model structures that would be relevant. The related question is kind of how you well, or if you had any advice or on how to better integrate the different areas within the Secretariat into the MSE process, in particular the FIS kind of survey function, the stock assessment function, the biological research program, and the MSE.
Wow, those are big questions. So to look at the first one, how would I prioritize any potential operating model structure?
Yeah.
I don't really have any advice to offer here. I think that it's working as is. Typically in these kind of situations, you want an operating model that's slightly more general than your stock assessment model assumes. So it allows for things like movement if your stock assessment does not allow movement. And what often happens is you keep on adding features to it, and eventually at some point you decide the underlying structure is not sufficient to support all these new features. Maybe we need to write a new operating model. I don't think that's that likely in this situation. So I think the current operating model structure is going to be fine for most uses. For the second question, how would I better integrate all the areas in the Secretariat between the survey and the biology and the MSE and stock assessment? I didn't really come across anything that struck me as being a problem there. So I don't have any advice on changes that are needed. But perhaps there's something specific you had in mind.
Well, I think one of the issues that that everybody faces is integrating these areas. You know, obviously people have a lot of work within their assigned function, but there's also a need, especially when you're developing an MSE like this, is to integrate across the different functions. And I think it's an ongoing challenge. That any advice would help.
Yeah, I mean,
it might just be that.
Tricky.
Yeah, it might just be something that is beyond the scope of a review of the MSE on its own. But that's fine.
I guess I would approach that slightly differently, and I would say that if there's a particular part of the process that's seen as not working well, so trying to think of an example. So for example, if it is felt that it would be better to have more of the survey analysis be incorporated into the stock assessment rather than doing the survey, analyzing it, and giving a final number that goes into the stock assessment. So should you integrate the survey into the stock assessment? And that would have trade-offs. So the stock assessment would be way slower, but it might more accurately represent all the uncertainty. So then you could run an MSE that says, okay, well, what if you did integrate that into the process? What would happen? Would it be more accurate? So you run the MSE for a certain number of years, certain number of simulations. You ask, do you end up with a more accurate final process? In other words, better catches, less catch fluctuations, higher biomass, or is it much the same? If it's much the same, then you would probably conclude that having a stock assessment process that takes 10 times longer is probably bad if it doesn't give you any benefits. So you could certainly think of scenarios to test almost any part of the process. They just have to be really well specified. And it's not going to be possible to effectively simulate through the MSE process the entire IPHC from gathering individual data points to analyzing surveys, to analyzing biology, to making political decisions, that's not going to be feasible in general. It's better to stick with specific requests.
If I just have one other question I want to ask, and so you mentioned in your presentation about exceptional circumstances. I'm not sure I recall that from the report.
There were a couple paragraphs hidden. I didn't highlight them.
Okay.
They are in the body of the report. And the general idea here is that this really comes down to when you use MSE to find a harvest control rule you're going to implement to set catches into the future. So it's becoming more like an automated process where every year you gather the data, get the survey estimates, you plug it into some harvest control rule, maybe there's an assessment involved in between, and that comes up with your catch for the next year and how it will be allocated amongst areas. So it's an automated process that doesn't involve a lot of haggling within each year. So in a lot of places where they've done MSE, that's what they want at the end process is every year, if the data looks like this, this is what your catch will be, and no sort of political haggling in between. Now, IPHC works a bit differently, so maybe that's not the process that would be followed here. But if that is the end goal, you need to have a way of escaping from that automated process when things go differently to what you expected, differently to what you modeled. So the example
so I think that's my specific question. That's my particular question is how do you know it's time to invoke something like exceptional circumstances or not?
Right, and that's why it's important to pre-specify them. So a classic example for IPHC would be, what if there was no survey for 2 years? Okay, and your rule for setting catches relies on there being a survey every single year. So now suddenly your MSE process that models a survey every single year does not model this particular situation. So that might be an example of an exceptional circumstance. And if that happened, you would invoke that exceptional circumstances rule, and you would say, okay, we cannot use the harvest control rule the MSE said was fine because the MSE wasn't conducted under the situation we have right now. So usually it's something that's major, or what if you discovered, like for southern bluefin tuna, we discovered that catches were vastly under-recorded. So for IPHC, the equivalent might be, what if you discovered that a fleet from an unnamed country had been fishing in Alaskan waters and the true catches were 50% higher than you thought they were? Then you might want to invoke exceptional circumstances. So it should be something that's major and out of the realms of the simulations you've done.
I think some of those you'd probably include, which it sounds like I mean the team should be maybe listening to this and thinking about these kinds of situations and include them in the simulations. These are kinds of things that you would want to be robust to as well.
Yeah, I mean usually in MSEs there's a distinction between regular things you might expect and the sort of unexpected things. And if you think something is very likely, then you should absolutely put it in the MSE and just run it so you know it works. But if you can't imagine that there is some reason why catches have been underreported by 50%, then there's, you wouldn't really think that was necessary to simulate that. But you might want to say, if we ever did discover that, we'd want to stop and go back and redo things.
Okay, do we have anyone else? We have quite a few people in the any of our observers or Secretariat team members have any questions? Okay, well, hearing none. Let me just canvass the SRB quickly, whether we need, anyone would like to have a private discussion with Dr. Branch.
I think we raised most of the issues that, or the questions that I have. Note that some of those elements we have already discussed. So I would like to congratulate Trevor for a comprehensive review of the process, highlighting the things that make it a useful tool and the need to develop this further or use it as part of the management toolbox.
Thank you. And if there are any specific comments, please do feel free to email me, and so I can incorporate that into my final report.
Thank you. And thank you for a comprehensive review. And yep, sorry.
Yeah, no, if I could just comment as well. So from the Secretariat's point of view, I think it would be useful that the SRB board members do have either an additional thought process and then potential exchange with Trevor on his draft report. Either today, tomorrow, or over the next intervening period, so that Trevor can get his final report to us. I think that would certainly be a good way for you to have a candid discussion about some of the findings or recommendations with Trevor offline. But we'll leave that to you and Trevor to organize, and we'll be in touch with Trevor as well. So thank you again.
Okay, thank you.
Thanks, everybody.
Okay, thank you, Trevor. We'll be in touch.
Okay.
I guess I'm looking at my agenda here. Okay, it's 10:30. We've completed the FIS, the stock assessment, and the MSE review. And I think we're ready to I think we're in the process of transferring our SRB draft report into a new template. So I think we'll wait on that. We'll let Dave do his magic, and we'll just go over those later. So let's get into the biological research program. We have until noon for this topic, so we have an hour and a half. Do you want to take it over, Joseph?
Yes, can you see the screen?
No.
Sharing?
Oh, yeah, yep. Yes, I see it.
Yep. Okay. So can you confirm that you see the screen?
Yes,
it's a bit slow, but I can see it now.
Yeah.
Yes, I see it too.
Okay. All right, thank you. Good morning. I'm going to be presenting the Biological and Ecosystem Science Research Update. This is agenda item 7, and that will speak to the document SRB017-08. So what I'm going to be doing in this presentation is providing very few updates and mostly focusing on responding to the requests and recommendations from the SRB in the past meeting, SRB016, and we thank the SRB members for providing that input that is really important to our process. Let me start with the first request in which the SRB requested an updated presentation on the plan and timelines for integrating research and results from the biological and ecosystem science research plan into specific functions and parameters of the assessment and the MSc process. We have, in fact, completed a first iteration. Not a first iteration, but a second iteration of this plant integration. The first iteration was the one that you saw and that was present in the document, and that's where we identified the 5 research areas on the left, the research outcomes on the second column, and then how those research outcomes are relevant for stock assessment and represent inputs for both the stock assessment and the MSc process. So based on this first iteration and trying to answer the SRB's request for improving this and specifying further the interactions between the biological research program and stock assessment and MSc, now we've come up with a revised integration of all these activities, and you can see this in this fairly busy table, in which again you have on the left the research areas, and in this slide you see from top to bottom migration, reproduction, and growth. The second column to the right is research activities, and the third column is research outcomes. The fourth column is relevance for stock assessment. The fifth column is relevance for MSc, and the last column on the right is specific analysis input, and that describes really how these research outcomes are going to be used both for stock assessment and MSc. So let's give you an example. For instance, if you start from the top, larval and juvenile connectivity studies, the research outcome is improved understanding of larval and juvenile distribution. Relevance for stock assessment is to improve estimates of productivity. Relevance for MSc is improve parameterization of the MSc operating model. And the specific analysis input is that this information, that research outcome, will be used to generate potential recruitment covariates and to inform minimum spawning biomass targets by biological region. So if you go down the list, and I'm not going to mention every single one of them, we have identified what are the specific analysis inputs for each research activity. So you'll see that the next research area in reproduction, there's a number of those, starting from histological maturity assessment, examination of potential spawning, fecundity assessments, examination of accuracy of current field macroscopic maturity, classifications, etc. All this are really relevant for stock assessment in scaling biomass and estimating reference points. They're relevant for MSc to improve the simulation of spawning biomass in the operating model, and each of them have specific analysis inputs that are described on the right. So this is just to provide you an example of how we're now providing or trying to provide further detail into how each of the research activities are related to specific analysis inputs. The next table includes the rest of the research areas, mortality, survival assessment, and then lastly, genetics and genomics. And here we're incorporating as research activities, the research activities that we're actually planning and contemplating in the next iteration of our 5-year research plan. So you'll see novel aspects that we have not yet touched on, but that are going to be included in our next 5-year research plan. If you look at the last research area, genetics and genomics, there's a mention on close kin marker capture studies, landscape genomics, and genome-wide shadon analysis that are all intended to improve estimates of productivity, the improved parameterization of the operating model, and that they have specific analysis inputs related to those. So in order to provide you with an indication of how we temporarily will approach all this different activities. We've revised the temporal chart of research outcomes, and you can see that the gray areas is where we actually plan on be working on this different research outcomes, and where you see the X is where that information we expect it to be available for stock assessment and MSD process. One clear example is for instance, in reproduction. You see the first one, histological maturity assessment. One of the research outcomes is updated maturity schedule. This is something that we're currently working on, and we expect to have an updated maturity schedule available at the end of 2021. Same thing with skip spawning. I'll touch on this later because this was one of the requests from the SRB. This is something that we've started to consider, so we've started working on it in the second half of 2020, and hopefully we can provide useful information by the end of 2021, and so forth. And that's what we've done for each of the research activities and research outcomes that were identified in the earlier tables. So, for instance, if you look again at genetics and genomics, the new studies that we're going to be planning on in the future, namely close-skin marker receptor studies, landscape genomics, genomic-wide association analysis. Those are planning to be initiated sometime in 2021, and the outcomes will be available 2023 and 2024, obviously depending on funding and depending on a number of considerations. So this was provided. Unfortunately, we did not have time to provide this in the document that was published, but this was provided in the presentations that you should have had for a few days now, and I hope you can comment specifically on how we've produced this temporal and integration analysis. One update that I would like to provide is that the study that we conducted on larval connectivity, looking at connectivity of Pacific halibut at early life stages between the Bering Sea and the Gulf of Alaska is now has been accepted in fisheries oceanography. That's Lori Store's paper. Yes.
Can I interrupt?
Yes.
Sorry to interrupt, but before we move on, looking at the details, maybe we should just back up and comment on those tables. Yeah. Rather than just wait to the report, I think maybe we can have a brief discussion of these now. Does anyone have a comment, question, input on these tables?
Well, I'd like to thank Joseph, and I'm sure this involved interactions among all groups, which is really encouraging that level of integration and communication is taking place. I'd like to take a look at it. This is just a skim through it, but it's really a nice extension of the appendix that was put into the report. I know a number of the things that have been going on in each of the 5 subsections has involved collaborations, but many of these, I think, the funding has ended and that sort of thing. And what strikes me is this is, the timelines is going to be a lot of work with existing staff. And I guess as Trevor had made the point that the MSE, the support for that program, given the workload anticipated in the coming years, might need to be revisited. I wonder if you could make some sort of statement regarding, your ability to meet some of these goals that you've outlined in the calendar. with the current staffing level that you have, and if it's going to be done solely with IPHC staff.
Yes, thank you, Kim, for your comments. Yeah, you're absolutely right. These are our tentative dates of producing these deliverables, and the topics are also tentative, and they're based on our ability to secure external funding. Our staffing right now is very limited. And we will require additional funding. So for some of these areas, and in particular, we're going to be working on prioritizing some of this work and some of these proposals. Some of those are going to be selected for external funding, and they will be exclusively depending on external funding. So I would like to stress, and if I didn't before, that some of this are depending on our ability to secure funds to conduct the work, and also secure funds to hire competent people to conduct some of these studies.
Josep, could I follow up on that? So you mentioned prioritization. Which of these activities are the highest priorities for the stock assessment? Which of these biological unknowns have the greatest leverage in the assessment?
I could comment on that, but I think that's a question that would probably Ian will respond more accurately than myself. Ian, if you can, could you comment?
Sure, thanks, Josep. I think there's a scale of different importance levels on these projects, some of which are going to be becoming more important over time. For example, the maturity, skip spawning, and fecundity information sort of begins as basic information, and the initial inputs into the stock assessment, for example, a revised maturity schedule, is unlikely to change dramatically our perception of stock status or any of our reference points. But once we have that and we start to move forward into tracking that information over time, if we start to see changes in maturity over time, or skip spawning, or fecundity, that's the sort of thing that could become very important right away. So what we did, and I guess it's not in this version, it was a previous version, we were asked, the SRB requested that we, instead of ranking all the proposals or all of the topics, the request was for us to just identify the top 3 in each category. And I guess we've kind of lost that here, although we have identified really only those projects for which we feel are a high priority in this initial list. So I suppose we could go back and actually try and rank these if you guys would like to see that.
That is fantastic, Ian. I think that that kind of ranking, or at least those top 3, would be really interesting to see, and I think would be a useful guide for prioritization.
Thanks. I think, sorry, go ahead, Sven.
I just want to echo that. To me, that's what this table was for or about, so that we could review the prioritization and make comment about what we think is there. The one element that's still missing is the likely cost in that, because obviously that plays into the prioritization. I mean, I see these as aims rather than objectives in many ways. It's not clear how you link the research outcome, how is that functionally going to happen? What is its likelihood of success, and what is its relevance, the scaling of the relevance to the stock assessment. So at least having your judgment on this prioritization would be very useful.
This kind of leads into the question, or what I wanted to mention. Can you go back one slide? Yeah, so this is a good one. First of all, I agree. I agree with what I think everyone is saying, is that this is really helpful. This is a great addition, and I think it's something that, you know, is coming into focus here as to what we're talking about when we talk about improving integration, and as kind of a living, you know, structure that could be carried forward and actually worked from. So I just want to express my appreciation on that. And I also, and one of the things we're hearing from everyone, including myself, is that so one thing to include here would be a prioritization, more explicit prioritization. That's one. The other is I think we just saw, or we just heard an example of what we've been asking for in terms of integration. So I forget who asked it, but I guess Kim maybe asked the question that Josep deferred to Ian, and then Ian gave the answer that is not in this table. So, you know, the relevance of skip spawning to stock assessment. Well, you know, if you can even go up a slide. Yeah. So very generically here, it's, you know, relevance for stock assessments to scale, biomass, and reference points. So that tells me that there really hasn't been an explicit internal discussion about this, and that can go in this table, but obviously it's there. Ian has a plan for this data that he just expressed pretty articulately as to what it's going to happen. So that's what we want in that box, rather than, you know, kind of generic improve this, improve that. It would be, you know, you could even have a bulleted thing of what the plan is for that.
Yeah,
I think that would help a lot.
Yeah, if I could chime in, it's David here. So we are actually going through that process at the moment and looking at revising the current biological research plan, because it ends next year. And the next plan will not only incorporate specifically the biological and ecosystem science research projects, but it will be an organizational-wide research program. And so it will obviously include those key priorities. And I think any feedback we can receive from the SRB at this point would also be very helpful. And then also sort of that delivery timeline of those key priorities. The overlaying factor, as Josep has pointed out, is obviously the finance side, but that's more of an internal rationalization with commissioners. But that's the intention. So yes, thank you. Any additional comments on the table at this point would certainly help us frame that new 5-year plan.
And so in the specific analysis input, you know, as I was looking at that, obviously I was thinking, you know, timelines, when is this going to happen? And then, you know, you went to the next, if you just flip down to the Gantt charts there, which also, this is really helpful. The suggestion I have here is to, and you guys were even talking about this a little bit, I think, this seems to be the plan for completing these biological research studies, I think, which is fine. I think what we also need is some indication of when these are actually going to be input to whatever the assessment or the MSc or the FIS designs, because it may be that the stock assessment won't be in a form or won't be able to handle that information when it comes in. Like, it may be that, you know, once that information is collected, it may be a year or two before it's actually implemented. So that, you know, just drilling down also into the specifics of the implementation, rather than just, you know, when the studies, you know, when the proposal funding, when the funding ends and when the conclusions and papers will be written. Well, that stops at the research, and I think should continue to be specific about when it will be implemented. So we can, I don't want to write major bullets in the report about this, but we'll try to summarize these things, but this is the intent of those comments that will come out.
I mean, has there been any attempt as a modeling exercise, either for the MSc or otherwise, to evaluate what your gut feeling might be in terms of the relative importance of each of these research activities in the different in the 5 different areas to the stock assessment or MSE?
This is Ian. I can speak to that. We have in fact included nearly every year in the stock assessment between maybe 1 and 3 or 4 sensitivity analyses specifically designed to address these topics. Until a couple of years ago, one of the major foci of that effort was the sex ratio at age. So I, for about 5 years running, included a sensitivity analysis in the stock assessment document to alternative sex ratios at age for the commercial fishery to illustrate how sensitive the assessment was and how important that new information was going to be. So what we've tried, and I've done that for whale depredation in the last few years. We've looked at differences in the maturity schedule and skip spawning. I did an evaluation of maternal effects. Actually, part of that was included peripherally in the document you guys saw in June. So that's exactly the approach we've taken, which is to take these priority research topics and explore them a little bit in the stock assessment prior to actually receiving any formal results from them. It's of course challenging because we don't know how important the answers are going to be. If we get an answer that's essentially exactly what we're assuming now, then it's certainly of less importance than it might have been. But we have used sensitivity analyses in the stock assessment as a way to quantify and also to prepare people for the potential for change as new biological understanding comes forward.
That's an interesting. I mean, that one way I can see this working, for example. I mean, some of these topics probably arose independent of the stock assessment. There might be some. Some of them might have arisen from gaps in the assessment. So as well, some are going to arise from gaps in the MSE, you're probably all of them. But it might be interesting to have kind of an independent prioritization, you know, from the stock assessment point of view, you may have some ideas about things that you know that this assessment is sensitive to that may or may not be included in the current research program. So it would be good to say, okay, here, if Ian had his wish list and this is what it would be in terms of research that's relevant to the assessment. Same thing for the MSC, same thing for the FIS. And then, you know, work from, you know, see how that compares to what is actually being done and how that, you know, priorities are. Okay. Do we have any further comments and inputs on this?
So would it be possible to see another revision of this table that would include a prioritization as well as, you know, a more fully fleshed out table with some of the research outcomes more fully developed in terms of application?
Yes, Kim, that's a good point. And this is a work in progress, and myself, Ian, and Alan are revising this constantly. So yes, we can produce an improved version, incorporating that prioritization and the application aspects that you just mentioned. Yeah, that we'll work on that immediately.
And I would expect, you know, obviously that's a request for next June. But Dave, can you just remind me, I don't know if you said it in your comment, but when is the organizational research plan scheduled to be done?
It expires, what, mid next year, Joseph? Yeah, so the idea is to try and draft that over the next couple of months, and then we'll be presenting that to the commission, potentially in draft format in November, and then also for their final informal endorsement in January. The commission doesn't necessarily have to endorse the report itself. It's an internal program based on requests and recommendations from the commission.
Okay, so I didn't catch that. So this research plan is going forward this particular year, and it's going to be presented for endorsement at the upcoming annual meeting or the next one?
Correct, because the current plan ends at the middle of 2021, so we want to get the new one in place so that we can work out our budgeting associated with it prior to the others expiry. So it will commence in the second half of 2021.
Okay, so by the time we see this again next June, it will have already been decided.
That's correct.
Okay, so I guess what we're going to do is provide some specific recommendations for ways to revise and improve this chart, and assuming they're going to be done and incorporated into that plan. So I think to answer Kim's question, unless we have some other work between now and late fall and January, we won't see this again. Which I don't think is a big problem. I think we'll trust that you guys will implement these and the requests that we make.
It would help to have a copy of this, this presentation that we could download and look at when we're writing up our notes on this section.
Yes, Kim. I thought you had these presentations already.
Should all be online.
They should be online. We will definitely check, but this presentations should have been online at least 10 days ago.
Yeah.
Yeah, we have them already. Thanks, Joseph.
Yep, here we have it. Okay, great.
However, if the SRB wants to edit directly the tables, I can provide the spreadsheets rather than just the PowerPoint. So please let me know if the Excel files that contain these tables would be more useful to you than just looking at the presentation.
I'll leave that to Kim if he wants to suggest edits to it. I think we'll decide that in when we move to that part of the report rating. I did just notice that our observers are muted. I don't know if that's intentional or not. Hopefully not. And so they did have some questions when it came to the MSE. So I think maybe what we can do, since Trevor's gone, is incorporate those into our follow-up work with Dr. Branch.
Yeah, if I could suggest, Sean, observers should be able to just raise hands, and then we can periodically check, and then you can call upon comment questions from them. That's going to be easiest. Otherwise, we have to leave open the meeting pane, which covers part of the screen for us, and makes it difficult to follow.
Okay, well, she apparently raised her hand, and maybe Anne-Marie, if you're listening now, you can raise your hand again and see what happens.
Yeah, just to make that clear, it's very difficult for us to track it in real time, and so if we could have when you break, if you then make a specific request for us to check to see if there is any raised hand from observers, we can do that. Otherwise, if we open that on the screen, the presenter can't present very well.
Right. Okay.
But the science advisors are now panelists, so they can ask any questions if they want to.
Okay, let's continue.
Okay, well, thank you very much for your comments on this section, and again, this is a work in progress that that will be continued and hopefully improved. So let me. Okay, so yeah, I just want to make a brief update to just let you know that the study that we conducted on larval connectivity between the Bering Sea and the Gulf of Alaska has finally been accepted for publication, and this will be appearing shortly in the journal Fisheries Oceanography. This is Laura Sadoro's paper that we've conducted in collaboration with the Alaska Fisheries Science Center here in Seattle with Janet Duffy Anderson, Bill Stockhausen, and Esther Goldstein. If we go through the different requests, request 11 from the SRB017 was related to migration and distribution, and you noted that the genetic data may be complementary to data collected using other methods. For example, stock structure at the genetic level could be reflected in individual differences in otolith chemistry if primary otolith annually are integrated. And then the SRB requested specifically that a portion of individuals that are selected for otolith chemistry also be used for whole genome sequencing. This is a work in progress. In fact, the proposals to address this issue are now being contemplated. So we don't have currently any research projects by which we can collect otoliths as well as genetic samples, but proposals are now on the table to address this issue. So research projects to investigate larval and early life stages, source, locations, and pelagic duration through otolith geochemical analysis combined with genetic analysis are under consideration. So this is something that we're working on right now to come up with a sound proposal that would integrate these comments from SRB. Are there any questions regarding this response to the request?
I don't hear any. It seems like a sensible approach.
Okay. Regarding the next research area, there's going to be a couple of requests that are related to sex ratios, and I just want to highlight that you know, we're continuing with the genotyping efforts that we started in 2000 with the 2017 commercial samples. We're genotyping now in-house approximately 10,000 fish per year of commercial samples. And one of the requests from the SRB was related to, obviously, sex ratios, but that was related to the use of otoliths to derive information from historical sex ratios. So the request was specifically that the IPHC Secretariat conduct a pilot study to determine whether DNA and PCR amplification of sex-linked single nucleotide polymorphism loci can be obtained from archived otoliths of different collection periods to demonstrate feasibility to develop a more comprehensive spatial and temporal sex ratio database. So we've conducted a little bit of work, and I'll pass the word to Anna Simeon, our biological laboratory technician, who will be updating you briefly on this issue. So go ahead, Anna.
Yeah, thanks, Josep. Yeah, so like Josep said, this is in response to the request to get some more information about, you know, the potential for using our extensive otolith collection to help identify past sex ratios. And we do have a few results here for you. And I'd like to preface this by saying that over the last, ever since the IPHC has been collecting otoliths, there's been just a couple ways that they've been stored. According to Joan, the Age Lab supervisor, starting in 2010 is when we started our dry, clean otolith collection. And prior to 2010, we basically have everything stored in a glycerol thymol solution. So as of right now, just in a real basic pilot study, so of those 7 otoliths that we were able to basically kind of simulate the storage of those that are, in the more recent years and from 2010 to today. So we were able to get the correct genotype off of the dry samples. The ones that are in glycerin, which represent the samples that are stored basically everything prior to 2010, so far haven't been successful in that. But with talking with Kim yesterday in a meeting and looking into the literature, I think we have some ideas as to how to get to some of those, how to get to some of those genotypes. So hopefully by the next meeting we'll have an update for you as far as that's concerned. And I think with those, what do we have in house now? Like 2 million otoliths in archives. We certainly hope we'll be able to get some out of the years that we're most interested for. There are a few complications along the way. For instance, like Joan has indicated that, in past years, some otoliths were cleaned in muriatic acid. There's no indication of which ones those are. So there's certainly going to be some that there simply won't be any DNA left to be taken. But again, I think we've got a good shot at getting the other ones.
Great, thank you, Anna. Are there any questions regarding this response to the request?
Sounds exciting. Hearing no comments. Look forward to next update in June. Thank you. Continue.
So moving on, one of the other research areas within the biological program has been to characterize the annual reproductive cycle in order to improve current estimates of maturity. This is a study that we initiated in mid-2017. You've heard before that in between 2017 and 18, we did an exhaustive program of collecting gonadal samples from both males and females during an entire calendar year from September 17 until August 2018, and that was to try to characterize the annual reproductive cycle and gonadal growth and maturation periods for both females and males. We actually collected 30 males and 30 females at every month all in one location, the Portlock area in the central Gulf of Alaska. This area was selected because it's a known spawning area historically. And the number of males and females was selected mostly based on the literature. I mean, this is a reproductive assessment study on that the main objective was to derive information for the first time in the species on reproductive development. Previous studies in other species doing similar studies that used between 10 and 15 females per month of collection. We increased that number up until 30 females and 30 males. That I think will be sufficient to provide with a clear description of reproductive development in the species. So what we expect out of these studies that are still ongoing is first, obviously, an accurate staging of reproductive status by conducting histological analysis of ovaries and comparing that with the maturity that is collected in the field through macroscopic or visual examination of gonads, to update maturity at age estimates, and to identify potentially skip spawning in this species. There's no information prior to this, at least from histology, and to start looking at fecundity. in this species for which there's no information. So one of the requests that the SRB issued in the last meeting was request 13, and that is that preliminary analysis of existing data on skip spawning be presented. And this is, we've started this work, but this is obviously linked to our understanding of reproductive development and oocyte development. In order to provide with information on skip spawning, we need to know exactly what are the reproductive characteristics of the ovary in Pacific halibut. So one of the first things that we did was to describe the oocyte developmental stages, and this study, a copy of which was provided to you in SRB017, and this paper is now published in the Journal of Fish Biology. So this is a paper called titled "A Comprehensive Description of Oocyte Developmental Stages in Pacific Halibut," and this is the base really for maturity staging that is based on histological oocyte stages. So what we do with this, in fact, is to stage females during the entire annual collection. So what you see here in this, and I think the animation played a little trick on me, but what you see here is on the x-axis you see months from March to the left to February on the right, and then you see the proportions of females on the y-axis, knowing that the total number of females in each of these months was 30 females. So what you could see is that when we classify the reproductive stage of females based on the histology oocyte developmental stages, then we can clearly see that beginning in March, continuing in April, May, and June, most of the females, probably more than 90% of those females, are beginning what is called secondary growth or vitellogenesis. That in July, a portion of those females are advancing further into what we call now secondary growth late stage in bright blue. And then starting in August and continuing in September, October, and November, then we start seeing those females developing further into full-grown stage of secondary growth. So that's late. That's what also is referred to as late vitellogenesis, and that continues until December. And what we see in January and February is finally females starting to get ready for spawning. So in January, that's when you first start seeing oocyte maturation and peri-ovulatory stage. So females with hydrated eggs just prior to spawning. And then in February, we see high proportions of females that show in their ovary show a high number of post-ovulatory follicles, meaning that those females have ovulated, almost 100% of them. And that coincides very well with what is expected in Pacific halibut in terms of the spawning season. So we see, and we can confirm that the spawning season, the peak spawning season in Pacific halibut is between January and February. So this information is crucial for addressing the issue on skip spawning, because now we can use this to identify those females that do not conform to the regular reproductive developmental pattern. For instance, here highlighted in this black circles, we identify females that during spawning and prior to spawning do not show or show signs of primary growth stage development. So females that are classified histologically as being well behind reproductive development. So we can look at the histology of those females. We can determine whether there are post-ovulatory follicles or not. We can also identify the presence of degenerating or atretic follicles or not. And then we'll look at the general structure of the ovarian tissue. That's comparing compacted ovary tissue ovarian tissue, which is characteristic of immature females, versus more loose ovarian tissue patterns that are probably more characteristic of females that have already spawned, mature females. So, and in addition to that, that is just histologically. We have the capability of complementing that information with additional reproductive and physiological parameters, and that's crucial for our ability, or that's going to be crucial for our ability to determine with a high degree of certainty whether females are skip spawning or not, and that is information on the gonadosomatic index, information on condition factor, information on fat content, but probably most importantly, endocrine markers that will tell us what is the, or confirm what is the stage of those females. And particularly, it's the gene expression levels of luteinizing hormone, a key hormone triggering the whole reproductive process, and blood steroids, particularly estradiol and a derivative of progesterone. So to give you an example of how we're looking at that, just at possible skip spawning females histologically, we identify one female, 1 out of 30 in November, that did not have full-grown ovarian follicles. This female was aged at 15 years old, knowing that the average half age of maturity is expected to be around 12. And the maturity classification based on histology was that this female was in primary growth and cortical alveoli stage. But the lack of post-ovulatory follicles and the lack of degenerating follicles indicate that this female has not spawned this year, but the loosely connected ovarian tissue could be indicative of this female having previously spawned in previous years, not in the current one. So this is a potential candidate for a skip spawner. But as I said before, we need additional information regarding the reproductive status of this female to definitely classify it as skip spawning. In the literature, skip spawning is very vaguely classified. Nobody has done anything else but histological analysis to classify skip spawning females. So this is in fact in the literature is still an issue in progress. In comparison, if we look at the females that were collected in December, one of the 30 females was in a very early developmental stage. That histologically, this female was classified to be in the primary growth perinucleolar stage. This female was age 9, and the age combined with a lack of post-ovulatory follicles, the compactness of the ovarian tissue clearly indicates that this is an immature female. This female has not yet reached maturity. So we have histological evidence to, or histological knowledge in the species to start looking at skip spawning. So this is a work in progress, and hopefully we can provide you with more information on the following meeting.
Question, Joseph?
Yes.
So could you remind me, do you have samples from across the range, or are these collected from a single location? I guess I'm asking because I was wondering if that pattern represented by your colored frequency histograms would be concordant across the entire range of the species, or if there's geographic heterogeneity.
Yes, thank you, Kim. Yeah, that's a really important question. This samples were collected only in that Portlock area. We only addressed reproductive development in at least temporarily throughout an entire annual cycle in one area. With this results, we can now start looking into the possibility of designing a study in which we could address spatial differences in maturity. And fortunately, what we know is that if you look at the months of June, July, and August, which is when the survey, IPHC Fisheries Independent Flatline Survey takes place, and that's where we could be collecting samples as part of our survey operation. Particularly those in July and August, we can determine that we know that those females that are collected, or at least most of those, will most likely advance. The Pacific halibut, as was indicated in the study that was just published and provided to you the previous meeting, is a species with determinate fecundity, which means that there will be, at the beginning of the spawning season, there's already a set number of oocytes that will be progressing. And that allows us to really use the collection time during our survey to particularly determine whether females will be advancing or not. But the question whether this is different in other geographic areas, this is something that we'll be addressing later on.
So if it's a determinate spawner, which means that the egg number is set, then the variation that you might see due to nutrition or other characteristics would be exclusively expressed as egg size, not egg number?
I would rather say egg developmental stage and the presence of atresia. Atresia is and oocyte degeneration is one of the characteristics of females that are either skip spawning or females that are under poor physiological condition. So I would say more than size, because the size range is fairly large. I don't know if you recall, there was a figure in the paper that was just accepted on the oocyte sizes based on diameters measured, and the range is fairly large. So oocyte diameter is not a good proxy for oocyte developmental stage. Oocyte size distribution, in contrast, is much more informative, but that requires a whole lot more work. So the presence of the most advanced oocyte, which tells you what developmental stage that female is, I think it's a better indication. So developmental stage is probably what we're going to be looking at. If there are no further questions regarding this request, there's another request related to reproductive assessment. That's a request for 14.
Sorry,
Yep.
Josep. Sorry,
Yes.
I was muted and I couldn't see my GoToWebinar panel.
Sorry.
Yeah, back one more. One more. The schedule there. Yeah. So two questions. I think one may have been answered already, but does this progression here account for age or not?
No, not yet. That is a good point. We do have ages for all these fish, and that's a work in progress. We're now looking at ages related to their developmental stage. That is, I don't have the results yet.
So this is,
but we do have the individual ages. This is not
so this is possibly showing the combined process of maturation of immature fish as well as skip spawning.
Correct.
Okay. So I guess the other question, which I think you had started to get to, but I was curious about the endocrine markers extension later on, and I assume that's what you mean by during this. I guess maybe not, but is the idea that during the survey period you could collect these endocrine markers and correlate them to proportion spawning, expected to spawn, to get annual estimates?
Not entirely. I mean, that is always the endocrine markers are complementary information to histology. I think the most sensible approach to assess maturity in a geographic or temporal manner would be just to limit yourself to histology during the survey months. But my mention on the use of additional reproductive markers, endocrine markers in particular, are to further characterize and confirm the reproductive stage of specific females. And that's when those markers are going to be crucial in confirming whether we have evidence of skip spawning or not. Those reproductive markers will most likely tell us with more certainty the actual reproductive status. The skip spawning, one of the major difficulties is distinguishing a female, at least histologically speaking, is distinguishing a female that is immature versus a female that is skip spawning. Because sometimes the presence of post-ovulatory follicles, which would exclude that particular female from having skipped, are hard to find. And depending on when the samples are taken, those post-ovulatory follicles, basically evidence of spawning at that season, degenerate over time, and you may not see them in histological section. So that's when I think those reproductive markers will come in useful to confirm the reproductive status of those females that histologically we're finding those as skip spawning or not.
Okay, so I guess I'm struggling to see how this is all going to be connected in the end, that because in your sampling period you don't really have, it doesn't look like a lot of predictive power for the upcoming spawning season as to what proportion are going to be spawning.
Well,
is that correct?
If you look at the months of July, August, and September,
mm-hmm.
you can see that in July we have maybe 2 females, the ones that I'm highlighting in green. Green is primary growth. Green indicates females that are underdeveloped, but they have not started secondary growth yet. So in July, you see that there's maybe 1 or 2 that are, that have to be examined. The question is to determine, according to their age, whether they could be immature or they're just slightly behind. But if you see August and September, 100% of those females, all 30 analyzed, all 30, in fact, are progressing through secondary growth. And in September, the same thing, with more proportion of females going from late to full grown. So based on this, you could deduce that all those 30 females collected in August, all those 30 females that we sampled are actually going to be spawning later that season. And the same in September. So the potential is there. Whether something happens to those females a little later on that causes skip spawning, that is something that we don't know. But at least looking at this bars here, this is indication that all those females, 100% of those females in August and in September, they have the capability of spawning that year.
Okay, so I guess and then thanks for that explanation. I guess what I'm trying to get at is ultimately, for this to inform the stock assessment in an annual spatial basis, you would need some way to accurately forecast, based on, say, August and September data, what proportion are going to be spawning in the time that you cannot observe in January and February. Right. So maybe I think that's what maybe another bullet or another follow-up to this would be, okay, how is all this going to work? So for example, you could take this data set and let's ignore the spatial variation because that's a whole nother ball of wax. Let's just say you were trying to do it for this particular location. You would collect all this data and then somehow analyze it to come up with a forecast model based on all these factors that you can use. But ultimately, the data that's going to be input to this model to generate the forecast will be these observations that you have in July, August, and September.
Correct, and that information on maturity, not derived from histology, but derived from visual observation of gonads, is collected in the survey, and that's the maturity information that Ian received. So that's one of the inputs into determining the stock spawning biomass is the maturity status. What percent of females that are staged during, June, July, August, September are mature based on visual observation. One of the outcomes of this study will be also to improve that staging and determine whether there's any errors in the visual assignment of maturity that is currently collected in our survey. Mm-hmm. Because there's a fair possibility that there may be some females that are misclassified. And this is a component of the study that we're still looking at, how good the visual classification of maturity is compared to histological classification of maturity, and how we can improve it.
Sorry to belabor the point. Go ahead. So you can continue.
Sorry, just a quick question from me. These were length stratified samples that you collected, or they're just totally random samples?
No, they were not length stratified nor randomly collected. There was a size limit, and we collected females that were at least 90 centimeters long. In order to make sure that at least during the spawning season, we will be collecting females that had that capability of being within the mature portion of the population. So all the females that we are looking at here are 90 centimeters or more.
Okay, so looking at this and making some assumptions about, how that's extrapolated spatially, presumably in the stock assessment, this would only change the estimate of, recruitment, which is also affected by other factors such as natural mortality and so forth, in the sense that it can't actually rescale this over time with further information. Is that correct?
I don't know, Ian, would you care to comment?
Yeah, I can. I'm not sure I'm following your question exactly, Sven. I think from the assessment perspective, we need to know what fraction of the spawners are actually contributing in a particular year. And we need to know whether that depends on their size or age, and potentially whether that's changing over time. And the last of those, I think, is the most important. If this is really just a case where only 5% or 5% of the stock doesn't spawn in a particular year, that's going to be mainly a scaling question, and it's probably going to come out in various scaling parameters in the stock assessment and have little effect on the stock recruit relationship or projections down the road. But to the degree that it does change over time and potentially over space, that's something we may need to account for. I think we're sort of getting a little bit ahead here. I see this as a case where, from the assessment, I need a car, and Josep has to start by inventing a combustion engine. So we didn't have any idea where we stood histologically in the summer. We didn't have methods to deal with this, and so this project up to now has really been just laying the groundwork so that we could even start to ask the right questions. And I think we're finally getting there, but we're not really able to ask them yet, given where we're at in terms of temporal and spatial sampling for this project.
And I totally agree. I think going backwards, and that was part of my question, you can't actually do anything with the data that you've got, or do you have the histological samples that you can reanalyze for this kind of information? That was the one thing. And the other thing is, if you are looking to see whether it's age or length or some sort of condition factor that impacts this, in order to be able to use what you have now, i.e. the length frequency information or the age information in your assessment, to figure out what the total or the relative change in fecundity given a kilogram of biomass would be. That was the thinking. That's why I thought this should have been collected length structured, ideally age structured, but length structured given the fact that you can't age them till later. That you could actually make an informed guess on the impact of lengths. So that's why I was wondering whether they're length structured. But I was just asking that whether that was done or not. Thank you.
I'll let Josep make the primary response, but I think that makes sense. And moving forward, as we start to develop a sampling plan for actually generating estimates going into the assessment process, that makes a lot of sense. At this stage, this project was really trying to, I believe, trying to optimize and get samples that were going to teach us the most about the process itself, rather than necessarily be inherently scalable in an upward direction toward population level estimates.
I guess I can see, if you were able to, these different colors represent a histological phenotype, and if you were able to get fish in where Josep has the circles, November, December, January, February, and you get phenotype green, then you have an idea that fish may be a skip spawner. But you're saying that you don't have access to large numbers of individuals to be able to do that. So you'd like to use the fish survey to do that. And so you're asking, in those earlier months, July, August, September, when the fish survey is going, given a histological phenotype, i.e. color, in a particular month, what is the probability of that fish being a skip spawner, right? In 6 months hence, that's what you're trying to do?
Yes, we have to recognize that, as Ian pointed out, this is the groundwork of trying to understand reproductive development in the species before we answer the big questions. And the sample size here is relatively limited. For instance, if you look at December, you may think that the proportion of, immature or possibly skip spawning females is really high. I mean, you see maybe 30% of those females in green, the rest are really advancing. Well, when we look at ages, and I have done that, all those females in December are probably all immature, histologically speaking, and because of their age. We need much larger sample sizes, and that's when it's going to come into a sound design that is length stratified initially, with a high number of fish, because what I suspect is that if there's any skip spawning taking place, that skip spawning, you will be able to identify, or you should be able to identify skip spawning females in August or September. In August, if you see, more than 90% of those females are at least on late secondary growth. Females that are skip spawning will not show that phenotype. That phenotype is for females that will most likely successfully spawn that season. And we see pretty much 100% of those in August, but those are only 30 females. So when we go back to the survey, knowing this, and knowing what to look for, at least in terms of histological signs of reproductive development, if we sample not 30, but we sample 3,000 fish, we know, we have now the information to determine, which proportion of those females may actually be spawning, skip spawning. and we can determine, what is the proportion of females that are most likely going to contribute to that spawning event later in that year.
Yeah, but I mean, conditional on the month in which the sample was taken, the probability of an individual having a histological phenotype, the color, is a function of its age, probably location, and a function of whether or not it's going to be breeding that year or not, right? And so that's a model, right? So you could develop a model, or think about developing a model that's looking at the sources of variation of this conditional probability of an individual being a histological phenotype conditional on month, right? So what Sven said earlier, having the age data associated with each of these fish with each histological phenotype, I think would be really important, and that would be one way of thinking about the analysis.
Mm-hmm. Yeah, I totally agree. That's a really good point, and that's going to be really feasible and informative when we have much larger sample sizes as well.
So the larger sample sizes, you are going to length stratify now? I mean, if you can just clarify from this, it looks to me like from your information that I've sort of kind of roughly interpreted as an ignorant halibut expert, you found one in the whole time period that was skip spawning, or that it was potentially skip spawning because it's not been fully documented?
Potentially skip spawning, and that's basically just one female that was 15 years old. When we see most of females 13, 14 years old being potentially mature that year. That particular female, 15 years old, that's the picture that I showed here. That female has all the histological signs of being skip spawning, but with our current understanding, which is limited, and without endocrine parameters, additional reproductive parameters that will allow us to confirm whether this is a potential skip spawner or not.
So this is 1 out of 360 females, is that right?
Correct.
So the scale of that.
Yes. Let me just add to that. Well, yes, it's 1 out of 360 females that we've analyzed. 30 females per month times 12. But the presence of that skip spawners in March or April, I don't know whether you would be able to identify those. So I'm not sure if that is really a meaningful conclusion to state that we only identified one female. I think the skip spawning period should be further identified, and we don't know when that happens. We don't know when that starts, when that signal could be detectable histologically.
I guess I understand that. What I was not trying to say that there was one. What I was suggesting is that one could reasonably conclude from the study that it's a very small proportion, or a small proportion of the population at that site that year.
That year with the number of females that we collected that year. That is a correct statement, yeah.
Okay, thank you.
Okay, we have 16 minutes to break, and what do you think, Josep? Will be able to conclude this section?
Let me try then.
Okay.
I think we should be able to do it within 16 minutes. Well, another request that the SRB, and this is really related to the discussions we've been having, is to incorporate size and age-specific fecundity in the next 5-year research plan. And that is, as you saw in the integration slide, fecundity was in there. We have not started these studies. There's quite a bit of planning to do before we address the issue of fecundity at size. Obviously, one of the objectives of this work is to establish that fecundity size relationship. And the measure is to try to determine potential annual fecundity as a measure of annual egg production. But there's a lot of important considerations that need to be incorporated here. And one is obviously the type of sampling, the time of sampling. In this particular species, we now know that in pre-spawning conditions, before females start the actual spawning, so prior to the identification of hydrated eggs, that will be a good time given the determinate fecundity pattern that this species has, but also the location of sampling and the sample size. So I think this is where one of the comments from the SRB to do that length certified sampling over time is going to be an important consideration. And the last consideration is the type of method. Now, in the literature, there are several methods described for fecundity. We have no experience there, but the choices are fairly clear. It's either gravimetric versus autodiametric methods. So depending on the technical difficulties involved, this is something that we're going to be investigating now. So what we're planning is into collecting some samples for method testing in 2021 during our Fisheries Independent Setline Survey, and then design a study to start implementing it in 2022. Are there any other questions?
I don't hear any.
Let me move on. There's really no updates on growth. We completed the study. We just finished writing the final report of the MPRB project, and this will be provided to you for the next meeting, hopefully with 1 or 2 manuscripts at least in preparation, submitted or published. The next area is fairly brief. We only have 2 slides here, and I'll pass the word to Claude to provide you with some updates here. Claude, go ahead.
Thank you, Josep. Good morning, everybody. I'm just going to give you a quick update on our DMR work. We've got essentially 2 projects, one that's fully underway, one that's kind of partially started. The first one, Josep, if you want to move on, is looking at improving DMR estimations in the longline fleet. This was partially funded by an SK grant. The work was conducted in the field in late 2017. And we've previously reported on some of the outcomes of that. We currently have a paper in draft form, primary author being Dr. Lohr, that looks at the production of a new estimate of anywhere between 4 to 8.7% DMR on these longline fish, which is consistent with our current estimates. We continue to do investigations into the relationships between the individual physiological markers, stress markers, environmental conditions, and the handling practices in relation to how those kind of map out to the release viability classifications. That's a component of a master's program that I'm currently involved in through the APU FAST Lab. Ongoing also is some work with the electronic monitoring. So the first steps showed that the electronic monitoring unit on board was very good at capturing which hook release method was employed to let the fish go. That involved either a careful shake, cutting the ganglion, or having the fish encounter the hook stripper, commonly known as the crucifier in the industry, on the order of 97% accuracy with that. We're now doing a little bit further work on both the EM video footage that was collected during this work, trying to use an electronic ruler to provide length estimation and see how accurate that is, to demonstrate whether EM can be used both to come up with the release method, for which we now have a kind of a fingerprint of injuries and release viability classifications that goes with each of those release methods, but also to estimate how accurate can we estimate the size and volume of fish that were exposed to each of those methods. So we've been going through the footage, looking at what the lengths are on a post hoc level on the work that we did in 2017. And we're also, we just recently collected some pre-calibrated camera lengths for the current fish season, just to see if we knew what the distance to the fish being released outboard the vessel with cameras that were pre-calibrated. How accurate is that compared to the post hoc one, where we had to estimate that from known kind of landmarks, as you see in the lower left there on the chute that the fish came up on. So that's where that work currently is, and we'll be doing some final reporting with the SK grant here between now and your next meeting. Joseph, if you want to move to the next one. This one is a project proposing to do something similar, but this time with the recreational charter fleet. That's the guided fleet. In Alaska, we have secured some NIFWIF funding to look at this. We started this last year with a questionnaire that was vetted by various personnel in the charter fleet, including some partners that are on the project, as well as some university staff up at University of Alaska Fairbanks, and Boudreaux, who's done this sort of questionnaire work with that sector before. We provided the questionnaire to charter skippers to look at their common gear practices, common gear employed or deployed, and the common handling practices seen within the fleet. There are some differences between practices or regulations for these fleets in Southeast Alaska, where there's a reverse slot limit, compared to the Central Gulf of Alaska area, IPHC area, regulatory area 3A, where they have a 2 fish bag limit, with one being less than a certain size. So there's no reverse slot limit there. So you see slightly different practices. We then want to move forward into a fieldwork component, somewhat analogous to the study I just presented, where we would go out and fish charter gear, either through option A here, where we kind of replicate the type of gear that was seen in that questionnaire. 75% of that was essentially circle hooks, and 25% of it was typically jigs, which typically have a J hook on them. And then also looking at the typical type of removals. So roughly 50% of the fish, they kind of reverse the hook back out through the hole it came in on. And on the other roughly 45%, they simply twist it back and around. They're pretty analogous removal treatments, pretty analogous to a gentle shake technique, although they're not always shaking them, they're just trying to twist the hook out by hand on small fish in particular. The second potential design that we could go out and test would be just to focus in on the most predominant methods, that being the circle hook and that reversal twist technique. And that would give us less of a confirmation on the overall DMR that might be applied to that sector, and more so specific best practices for one of those methods. And this work's also the subject of ongoing efforts to secure sufficient funding to try to secure enough pop-up satellite tags, those archival tags that we've spoken about before, that give us a pretty good estimate and are the basis of that previous study by Dr. Lohr that I mentioned, where we can get direct reports back from those tags roughly 96 days out that allow us to get a estimate on whether the DMR is currently being used by that to estimate that sector's mortality rates, whether they're in the realm of the reasonable or not. No work has been done directly on the recreational fleet before. The current estimation methods are based loosely on the commercial discards, and their mortality rates, as well as some other species such as fish on the East Coast, and striped bass and studies like that. That's kind of my update for now, where we're at with those two projects.
Claude, can I just ask you a little bit about the tagging here? So have you investigated the cost benefit for using a couple other tag technologies like acoustic arrays around the release location, or just plain old depth archival tags? I don't know what the recap rates are like, but if recap rates were high enough, those might be cost effective.
Roger. We have done some internal work on kind of a power analysis of how many tags might be needed to get within a range that we are interested in. But as far as the cost benefit on the different tag technologies, we did not investigate that. We focused on the accelerometer tags that show us whether the animal is actively moving or not. And those tags, because they're programmable and they can pop off, we get a much better reporting rate on those, and we don't have to wait as long to get that sort of information back. So like I was saying, we can have them programmed for 96 days, and we get quite a good, certainly at least in the long line study that we did, we had a very good reporting rate on those tags. I think Dr. Lohr's on the line. He could probably speak to, you know, what our recovery rate was, but it was quite high for those. Much higher than the wire tags that we put in most of those fish, where you have to wait for many years, and you're also dealing with those aspects of what's the reporting bias that we're also seeing here.
Sure. The pop-up tags are a few $1,000s a piece, though, right?
That's correct. We're looking at roughly a price tag of around $2,500 for those tags. So it's not insignificant, and it certainly limits how many fish you can tag with that technology.
Yeah, it might be worth just figuring out what it would cost to put a big enough acoustic receiver array in. You know, the acoustic receivers are not cheap, but they can often be borrowed or rented. But that brings your cost per tag down to about $300. So once you've got the array in, then you're much less limited in terms of the number of tags you can get out, and that would also give you very high reporting rates and data as soon as you pulled the receivers to download them. I think whether or not it works out, I think depends on how far they're moving after they're released. But you can probably get the same quality of information at the same speed. It's just a question of whether you can feasibly build the array large enough.
Thank you.
And this section was in the SRB017 documented as well. And first of all, congratulations on getting a new grant for this. I guess I was a bit surprised, given at least what was provided in terms of methods in the 08 document, that there was really not a lot of information provided so that the SRB could provide comment. There wasn't any information provided about how, where, how many fish would be collected, how the data were to be analyzed, what the sample size were relative to the variability in the data, and there were no statistical model provided to evaluate the efficacy of the project design. So, and further, I seem to remember from previous meetings that this recreational mortality was a really fairly small part of the overall mortality relative to the commercial harvest. So, the question is how relevant is the work to the stock assessment? So, this is another opportunity that the SRB could have contributed, I think, to this if we had opportunity to interact with, the biological section a little bit more in their design.
Yes, Kim,
Yeah.
this is Giuseppe. Yeah, the point is definitely well taken. Yeah, we apologize if not enough information was conveyed in there. The project I must admit was a little bit put on the not on the back burner, but it was conditioned to our ability to provide external funding for the completion of this study. So sample sizes, what we did, and that is something that we should have included, is that we did conduct a power analysis, a statistical analysis to determine, what would be a minimum number of tags that could provide us with meaningful results, and that was done, and that was included in the original grant application that it was submitted to the National Fish and Wildlife Foundation, and that was eventually funded. So yeah, I apologize for that. There was a statistical analysis conducted, and thinking back, I must have omitted including the grant application. I remember doing that specifically for other grants, but this one may have escaped me.
Okay,
I just wanted to comment with regards to Olaf's point, or some clarification about these acoustic arrays. And it notes that one of the question here is regulatory error or spatial variability in mortality, and I think that maybe you'd need more than one array, you'd need multiple arrays, and I think that may be the problem, but just wanted to seek clarification from on that.
Yes, this is something that this is Giuseppe again. Yeah, thank you for those comments, and for the alternative ways at looking at dispersal after release. But we did experience a very large movement of those fish that were released in our long line study. I'm not sure if it was on the report and presentations of the last SRB meeting, but this was presented to you before. We had a map with the dispersal trajectories of those fish that were tagged around the Portlock region that you saw in the research presentation. So this were tagged in the central Gulf of Alaska, and there were individual fish that move considerable distances. So I'm not sure whether that acoustic array would be able to capture the amazing movement that those fish experience once released. That I just wanted to make a mention of that probably the large movement capabilities of those fish once released would make that approach slightly difficult.
Okay, we're at 12:03. Joseph, how much time do you think you need?
We have, in addition to this, we have 5 more slides. So we could either break now and resume later, or continue. Either way.
I mean, we've been going for a while, so my suggestion would be to break now and continue after lunch. But on this particular topic, while we're spitballing ideas for improving these estimates, a few years ago, I worked with one of my PhD students, and we were testing the idea of a mobile array, where instead of setting up a fixed acoustic array, you attached receivers to commercial fishing gear, and then you would have a mobile array that could track pretty much any scale of movement, and you would get kind of almost real-time data coming in as to where those fish are moving. So if anybody wants to talk about that further, I'm happy to, or anyway, I think that's a distinct possibility that could possibly be investigated in the future.
Yeah, I'd just follow up on that. You know, I've done a lot of this sort of work, discard mortality estimates as well, particularly on the analysis side. And I think the SRB could definitely get involved in trying to help advise on this study, but it would require some more details than what's here. But I'm very interested in following up more as well, if that would be helpful.
Okay, so maybe what we can do is add an agenda item to the June meeting, perhaps, to get into this a bit more. Okay, so I propose that it's 12:05 and pretty close to on target here. So why don't we take a break until 12:35 and we'll come back and wrap up this section and then move on. Hearing no objections, let's do that.
Okay, thank you.